we are SulFind, representing the Norwegian University of Science and Technology in the iGEM 2021 competition. When the NTNU iGEM team first was assembled in the winter of 2019, we leaped right into discussion about the kind of impact we wanted to make with our iGEM project. It soon became apparent that we were all passionate about the environment and that we wanted to use our project to tackle local issues that affected the environment. Living in Norway, the world's second largest exporter of fish and seafood, we quickly knew that we wanted to contribute to solving a problem in the fish farming industry. Fish has been an important source of food throughout history, and in the last 50 years the consumption of fish and other aquaculture products has doubled. It is estimated that the world population will reach 9.9 billion people in 2050. Because of this, the fishing sector will be vital in the years to come. Fish are energetically efficient to produce, yielding more meat per unit of feed in comparison to other land-based meats such as cow. Because of this, the aquaculture sector will be often championed as a solution to feeding a growing population worldwide. This is also true in Norway, where the government plans to increase the growth of the aquaculture sector within the UN Sustainable Development Goals for Sustainable Growth. However, there are many high-profile issues that the aquaculture sector must overcome in order to support an increased market demand.
During our research and brainstorming phase, we came across recirculating aquaculture systems, or short, RAS. RAS are almost fully closed circuits where water is recirculated. As a result, most components such as pH, temperature, and oxygen levels can be strictly monitored and controlled. The problems of fish escaping and salmon lice are also non-existent in RAS. This governs fish welfare. In addition, no waste products from the facilities are released into the surrounding environments, as the system is closed. This ensures secure and sustainable waste handling compared to sea-based cages. However, the RAS sector is facing the problem of hydrogen sulfide toxicity. Hydrogen sulfide, or H2S, is a colorless gas that is produced by microbes, more specifically by anoxic sulfide-reducing bacteria. The gas is extremely toxic even at low concentrations. The presence of H2S in RAS has led to acute fish death on several occasions. This means massive loss of fish as well as great economic loss. So, how could we contribute to solving the problem of hydrogen sulfide toxicity? As part of our human practices work, we met with several stakeholders in the fish farming industry. They informed us about existing hydrogen sulfide sensors on the market. But these were costly and not sufficiently sensitive to detect H2S at levels toxic to fish. The industry expressed that a cheap sensor with continuous measurements and higher sensitivity was desirable. As a result, the goal of our iGEM project was to make a biosensor that meets the criteria expressed by the industry. Presenting SulFind, a biosensor for sensitive hydrogen sulfide measurements in recirculating aquaculture systems.
Several approaches were considered when developing a biosensor with adequate sensing capability for detection of hydrogen sulfide. Considering that H2S levels in the water tanks can rise quite quickly, one of the most important sensor qualities is rapid and sensitive detection. After many hours of research, we decided to develop two different approaches, a whole cell sensor and a molecular sensor.
When looking for target molecules for the sensor, we started by looking into bacteria with sulfur metabolism in hopes of finding a protein or mechanism related to H2S binding or conversion. We quickly became aware of an enzyme acting in the sulfide oxidation process in the bacteria Rhodobacter capsulatus. This enzyme, sulfide quinone oxidoreductase, or SQR, catalyzes the first step in hydrogen sulfide metabolism, where sulfide is converted to elemental sulfur. We started investigating how this enzyme could be utilized in a biosensor and learned that expression of the SQR gene is repressed by a repressive protein. When H2S is present, it is converted to elemental sulfur by the SQR enzyme. The elemental sulfur further binds to the repressive protein, making it disassociate from the operator and inducing expression of the SQR gene. Our whole cell sensor is based on this repressive protein. as it will directly interfere with present H2S molecules. In our design, a fluorescent protein named Krelov is placed under the control of the SQR promoter. When H2S is present and binds to the repressor, the Krelov gene is expressed and fluorescent protein can be detected. We have developed several alternate designs with different fluorescence proteins for both aerobic and anaerobic expression, as well as promoters of different strengths.
We were interested in developing a molecular sensor because it offers immediate and selective detection, and in our initial research phase, we investigated several approaches to using heme proteins as a sensor molecule. Heme proteins have a prosthetic heme group containing an iron atom that can bind gases like oxygen, nitrogen monoxide, carbon monoxide, and most importantly, H2S. This interaction is non-covalent and reversible, which offers a possibility for continuous detection. The interactions with H2S vary between different heme proteins, and our research revealed myoglobin as a promising protein for the sensor. After deciding to use myoglobin as the basis of our sensor, we needed to find a way to create a detectable signal for when H2S binds to the protein. For myoglobin, this is possible using the principle of first-order resonance energy transfer, also known as FRET. This is a method commonly used to study protein interactions based on an energy transfer that occurs between an energy donor and acceptor, usually fluorophores that are in close proximity. This energy transfer produces a fluorescent signal that can be detected. In our sensor system, we can use FRET to detect H2S binding because the absorption spectrum of myoglobin shifts when interacting with H2S. The particular method of energy transfer we implemented is called absorption into fluorescence and is achieved by labeling the N-terminus of myoglobin with a fluorophore that has an emission peak corresponding to an area of interest in the absorption spectra of myoglobin. When exciting the fluorophore, energy from fluorescence will be absorbed into the myoglobin, and therefore changes in the absorption can be detected by fluorescence.
We worked with both alternatives in the lab and ended up not moving forwards with the SQR approach due to time constraints. Therefore, we only present the results for the heme approach in this video. In the presented graph, we see the results of one of our first experiments. As expected from the theory, our null measurements were the highest. Additionally, there is low overall variance and a statistical significant difference between the null measurements and the first H2S value, 0.1 micromolars. However, the general trend is not perfect. Due to the unwanted upwards trend towards the higher values, we speculated that the large molar excess could be oversaturating the reaction mixture. Therefore, we tried to use a higher mixture concentration and narrower range of H2S concentrations. As you can see in the following graph, this did not go well. We got very high variance, no discernible trend, and the zero measurement wasn't significantly higher than the measurement with H2S. Following these experiments, we tried investigating different protein concentrations as well as the effect of time. In the graph shown, we see the Fi of several protein concentrations measured with 3 parallels at 3 time points. As evident, time does not seem to be a large factor in this experiment. To investigate the reaction with H2S further, we performed a series of kinetic experiments. Here we see that in the very beginning there is good separation between the H2S Fi and null Fi, but this shrinks over time in several of the graphs. In graph A, with the shortest measurement intervals, we see a much sharper decrease in fluorescence for both lines compared to the other intervals. In conclusion, although we have some results indicating the potential for an H2S sensor with a very low level of detection, the variance and lack of trend in important ranges severely inhibit the sensor performance. We speculate that a lot of the variance could be explained by the free diffusion of H2S from the open wells into the air inside the fluorescence measurement instrument. To diminish some of the factors affecting the variance, the system was implemented in a microfluidic system.
Next, our biological detection system was embedded on a microfluidic chip that can be integrated into existing systems for testing water quality in RAS facilities. Our sensor would be a LOC with a flow-through solution. In the chip, myoglobin is encapsulated in water droplets that are separated by a spacer oil. The same goes for our H2S solution, but in a different pipe. The two separate pipes are then merged, mixing the myoglobin with the aqueous H2S solution. H2S and myoglobin will then react, creating fluorescence. The droplets will be collected and then measured off chip. Ideally, such a full-on microfluidic chip allows rapid detection at a very low concentration and is a more user-friendly sensor.
The biggest safety risk associated with our project came from hydrogen sulfide. We produced hydrogen sulfide in solution by mixing sodium sulfide with water. This may lead to hydrogen sulfide evaporating in gaseous form, which is highly toxic even in low concentrations in the air. It was therefore very important that all experiments in which hydrogen sulfide is produced were performed under a fume hood or in controlled isolated systems. Sodium sulfide as well as hydrogen sulfide in solution are both highly toxic and corrosive and need to be handled with care. In order to handle hydrogen sulfide and the rest of our laboratory work in a responsible and safe way, we sent in safety forms describing our planned work to be reviewed and approved. In addition, we attended a small safety course, took an exam in lab safety, and attended a lab safety tour before getting access to the lab. From the safety training we got before entering the lab, we learned about the guidelines, methods, and equipment useful for mitigating the risk we previously identified. These include the use of fume hoods when handling hydrogen sulfide, never working alone, inactivating GMM waste through the use of an autoclave, and the aseptic technique. These were the most important safety procedures in our laboratory, as we did not use any other toxic chemicals other than H2S, and the bacterial strains we used belong to risk group 1, meaning that they do not cause disease in healthy adult humans.
In our education and communication work, we developed a learning activity game, visited a local high school, attended 2 conferences, and received publicity by several Norwegian aquaculture newspapers. We also reached out to the public by posting regular updates on our project through our social media platforms.
Build-back is meant to illustrate that bacteria may be engineered by taking up plasmids, and that these plasmids can be created by combining genes of interest. The game includes 3D printed gene pieces that are designed so that 4 pieces fit together to form a circle. The students can then build their own plasmid with 4 genes to engineer their bacteria, giving you traits that will help it thrive in a given environment. In September, we had an exhibition stand at Researchers Night at NTNU. Researchers Night is a part of the Researchers in Europe 2005 initiative, and is a yearly event at NTNU. The purpose is bringing research and the general public together through edutainment activities that increase the public's knowledge of research. The event is mostly attended by high school students and is a platform for promoting careers within research among young people. Acted with high schoolers about biotechnology, the field of synthetic biology, and how we apply synthetic biology in our iGEM project. We attended the Aquanor conference in Trondheim at the exhibition stand for NTNU Oceans. Aquanor is the world's largest aquaculture technology exhibition, and it's an important meeting place for presentation and discussion of innovation within the sector. At the stand, all our team members engaged with aquaculture companies and projects and discussed the potential of synthetic biology within the aquaculture sector. In September, we visited students at the entrepreneurship course at a local high school called Torastorm, and we led one of their science classes. We were invited to give an introduction to the world of DNA and synthetic biology. We also spoke of iGEM and gave some insight into how we decided on solving the H2S problem in RAS facilities.
We believe that taken further, our project SulFind will contribute to reducing acute fish death in RAS facilities, ensuring that resources are not wasted in fish production, and making RAS a more sustainable and profitable choice for fish farming. With this, we hope to encourage the use of RAS in aquaculture so that the industry does not grow at the expense of the environment.